We begin today talking about the tryptophan operon. Tryptophan operon is an operon that is under negative control, so we can tell by the presence of a repressor protein here. Let's look at the structure of the tryptophan operon. We have a promoter to which the RNA polymerase will be binding to. We have our RNA polymerase here. And once again, we have structural genes. And as we already have discussed over and over again, an operon has structural genes that are under the control of a single promoter. And in the case of the tryptophan operon, we have 5 structural genes which will result in the transcription and then translation of 5 different structural genes, each of which are enzymes that will result in a pathway. And eventually the pathway will produce the amino acid tryptophan. So E. coli has the ability to make its own amino acid tryptophan. And this is very important. That means you don't need to supplement the media that E. coli is grown in with that particular amino acid. It can produce it itself. And this is very important. It means that this particular operon is usually found in the on position. It's usually on. And so when the repressor molecule is made, it is usually inactive. And that's very important. It's inactive. It means that RNA polymerase binds to this promoter, and the template strand is read. We make messenger RNA, a polycistronic messenger RNA that is translated. We make our gene products here, and then the pathway, we have all of the different enzymes in the pathway, and tryptophan is made. When enough tryptophan is made, then we are then in a position to regulate this operon, and that's what we're going to talk about right now. So we've been talking about the tryptophan operon. E. coli needs to make tryptophan to live, but if tryptophan is available in the media, several things happen. Now, the repressor molecule is usually found in the inactive state, and that's because you want to keep making tryptophan. But when there is enough tryptophan around, or E. coli is transferred to media that contains tryptophan, this is what happens. So the first thing we need to remember is that molecules that interact with the effector molecule, in this case for the repressor, those are known as modulators. Now, in this case, the modulator is called a co-repressor. So our modulator molecule here is tryptophan. So tryptophan is the co-repressor. And what it's going to do, it's going to interact with the repressor molecule. In this case, because it's tryptophan, we no longer need any tryptophan, do we? We don't. And so it's going to interact with the repressor molecule, and it's going to change its shape so that it is now active. And because it's active, it is going to be able to bind to the operator. And once it binds to the operator, when the RNA polymerase binds to the promoter, we are no longer going to get any transcription, we're no longer going to get any transcription of our structural genes in the tryptophan operon. So even though we get binding to this strong promoter, we are not going to get transcription of our structural genes. So we have effectively shut down the tryptophan operon. So the tryptophan operon is a wonderful example of an operon that is under negative control that is repressible.